This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 958, recorded on November 25th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon DePalmieri.
Hello, everybody. And we were expecting rain here today, but we didn't get any. And the temperature is somewhere around 46 to 48 degrees, as I recall, having stuck my finger out the window. And it turned out to be a rather pleasant day. And yesterday, which was Thanksgiving, of course, everybody knows that, the Macy's Thanksgiving Day Parade went off without a hitch. And all the balloons stayed in New York, which was good. And the ballonatics, the people who actually hold them down, they're called ballonatics, by the way, they didn't have a very difficult job yesterday at all. They enjoyed themselves, and everybody had a good time.
It's 11C and sunny in New York, and New York is packed with tourists today.
They are.
This is the day after Thanksgiving. They all come in.
That's right.
Do stuff.
Yeah,
so,
yeah. They buy.
Yeah, it is. The trains were packed, and it's a holiday schedule on the train. Oh, well. Also joining us from not from Madison, from somewhere upstate New York, Brienne Barker.
Hi, it's great to be here. It is 40 degrees Fahrenheit, 5 Celsius. No snow coming down, but plenty on the ground from previous storms. And it's a good holiday.
Yeah.
From Western Massachusetts, Alan Dove.
Good to be here. I've got 9 Celsius, 48 Fahrenheit. It's a gray, overcast, cold. It was kind of drizzly, but I think it's just kind of misting now. Good day to just sit around and eat leftovers.
Good day to have a podcast, I think.
Yes, that too.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi. It seems to be the same as for Alan, at least in Celsius, where it's 9 Celsius, but my weather app says it's 50 Fahrenheit, so whatever. It's sunny. It's pretty nice out.
Today, for the first time in a while, we do not have a guest. We're going to do some papers. Yeah.
Yay.
We're looking forward to this episode.
We're going to TWiV like it's 2008.
We're going to TWiV like it's 2008. Yes, I love it.
Like we did last summer.
Oh, yes. We love our guests, but we also love our papers as well. So, hey, it's the papers that got us going in the beginning.
Yes.
So, Dixon, you and I started this in 2008.
Yeah, you started this, and I had reluctantly joined you, and I regret being reluctant because look at all the fun we've had. And
how many years ago is that?
That's a long time. That's 2008 to 2000.
I can't do the counting.
22. Well, yeah. Well,
18 would be 10 years, and then 4 more would be 14. So we just passed our 14th year.
14 years.
How about that?
Wow. I love it. No, I love it.
You weren't so reluctant, Dixon. You actually...
Well...
I remember I met you on the street walking to the garage, and I say, I have this idea for a podcast where we just talk about viruses. We just have a conversation. And you said, let's call it This Week in Virology, which is the title I had in mind. Yeah, I had the same title in mind. And so for the first episode, I think I came to your office, Dixon.
You did, and we forgot to turn on the recorder. I remember that too.
I did forget to turn on the recorder. I had all kinds of technical problems.
It was fun.
14 years ago.
And to this day, 14 years later, in, 18-point red at the top of the Google Doc we work from. Turn on the recorders. It's become plural now.
Sometimes I don't. And they are running, yes?
Yep.
They're all running.
Good. I see the red dot.
Hey, we're running in the browser.
I have a bunch of announcements for you. And I'm still having a blast doing this. This is a lot of fun.
Oh, yeah.
It's just great to talk about science. We have a bunch of announcements for you. First of all, we continue to have support from vaccinated.us. They're selling Spike T-shirts, SARS-CoV-2 Spike T-shirts. They're very nice T-shirts. They're made of nice material, and they have a three-dimensional image of the spike protein on them. Different colors, different sizes, and they are giving their profits to us through the end of 2022.
If you use the right code at checkout, right?
Yeah, you have to use the promo code MicrobeTV at checkout. I feel bad for Matt because he says he has to do a lot of manual correcting of people who don't do this or that. And he said it's getting busy now in our sales, so I don't have a lot of time to do that anymore. So, but he's going to do it through the end of December. So go to vaccinated.us and pick out your shirt, put it in the cart when you check out MicrobeTV. Just one word, no period. MicrobeTV. It'll tell them to direct the actually, now it's part of the profits. It's not all the profits anymore. After September and October, he said we need to have some of the profits going back to us.
They need to pay the rent, yeah.
Yeah, so that's fine. We're happy to split it because we have gotten very nice support from them so far. Everywhere I turn, people say, I have Spike T-shirts. They love them. Someone in the stream the other night said, I just bought 10 to give out for gifts.
Wow.
Nice.
Which is cool. I mean, it doesn't have any Microbe TV branding on it, which is too bad. But maybe that's why they like them. I don't know. Anyway, thank you to vaccinated.us. And we had an email from Robin who writes, I was anxious about ordering the woman's soft T-shirt. My worry was it would be sized for juniors and be too small. At my age, I don't want any shirt sticking to me. I don't either. I don't like tight shirts. No. Yes, I think you have to be young. Yeah, so for anyone else with T-shirt anxiety, I ordered what the chart said, and it fits nice. Okay. T-shirt anxiety. Another thing to add to your anxiety, if you don't have enough already, T-shirt anxiety. Okay, so then I want to remind everyone that we have 10 copies of Principles of Virology signed by all 5 authors, and if you'd care to donate $5,000 or more, I'll send you one. Just send me an email to Vincent@microbe.tv. I only mentioned...
By the way, I want to add this. Any part of this book, if you buy it and you don't understand it, Vincent will be glad to sit on the telephone and tell you all the details as to what it really means.
Operators are standing by.
Operators are standing by, exactly right. Don't call me, by the way.
If nothing else, he can tell you which episode it's discussed on.
Yes.
That's right.
If you want to donate, you can have a professor, absolutely, to explain things. Maybe not on the phone, but by email. That would be fine with me. So it's interesting. I mentioned this once before. I got no responses. So maybe the 5,000 is a little bit of a high price point. I understand.
I bet you could get lucky. You could get lucky.
Well, I'm patient. But what's funny is that people said, hey, I gave 1,000 last year and I never got my signed book. So I had 5 people write in. So, okay, I have to now get out. And these books are sold out at the publisher. You can't order them. So I'm digging around Barnes & Noble, Amazon, Abe's, and all those places to get up. And I'm getting copies. So those of you who have written, you'll get them. And finally, Miguel asked us to share a job posting. Miguel is with the Harris County Public Health Department, which is in Houston, Texas, with the Mosquito and Vector Control Division. And they have a opening for a testing and evaluation specialist. You're going to assist with and conduct projects associated with insecticide resistance management by testing various materials and products used in surveillance and or control of mosquitoes and other disease vector species. So put a link to that in the show notes if you're interested. Check it out.
It requires a high school diploma, and they'd like some microbiology lab experience. So if you, probably some listeners fit that.
And now, unfortunately, we have some sad news that Kathy's going to tell us about.
Yes. My faculty colleague in microbiology and immunology here at the University of Michigan, Octavia Fuller, passed away a week ago today on November 18th after a short illness that was not COVID. She worked initially here as a faculty member on herpes simplex virus, doing things on entry. She had done her postdoc with Pat Spear. Then she kind of transitioned into doing work with HIV in a program that she called Trusted Messenger Intervention, where the idea was that she would go to Africa, and eventually she took students from her class to Africa And they worked with clergy to teach clergy about HIV and what you needed to do for prevention and so forth. And the idea is that because the people will trust the clergyman, they would trust that messenger more than some person coming from the United States to tell them how they should behave and so forth. So there was quite a bit of success, and she got support for that. She also had an appointment for a while here in the Center for African American Studies. But most recently, her big claim to fame was that she was on the vaccine advisory committees. So those started, as you know, 2 years ago. December 2020 is when we really started hearing about them with respect to the new mRNA vaccines. And for the people on those committees, it was a lot of work. They had lots and lots of reading and preparation to do, and sometimes in a very short time. So they pretty much had to drop everything in their life and do all that. And then she continued to serve on those committees. So it's a really big loss. After she, or concurrent with that vaccine advisory work, because that was ongoing as additional things came out, she did town halls and lots of outreach. And one of them was a long form interview with someone from the Detroit TV station. And that person had lots of material and is featured on about a really nice 5-minute tribute that we'll put the link into. And it's also at asv.org at the In Memoriam section. So if you want to read about a really interesting person who took her virology in a different direction, read about Octavia Fuller. But we will really miss her.
Yeah. That's a huge loss.
Yeah, it worked out that I was on a couple of the ASV town halls and things with her. There were at least twice that I could remember that we ended up doing outreach together, and she was really wonderful at it. She will definitely be missed.
All right, on to the literature, as they say over at the PUScast. First paper is Nature Microbiology. Divergent SARS-CoV-2 variant emerges in white-tailed deer with deer-to-human transmission. And these are, where are these from? Somewhere in Canada, right? Yes, because it's a study out of Canada. They don't have the affiliations on the front page. It's at the end because it's too long. But we have Bradley Pickering as the first author, and then we have Samira Mubareka and Jeff Bowman as the last two authors. I don't know if there are any. Did you
pick this paper because I was going to be in upstate New York?
Or because I'm in Michigan and they features Michigan.
Michigan comes up in this, yeah.
No, I just picked it because I thought it was cool, but it's relevant, yeah. And we've talked about this before, this discovery, this knowledge that SARS-CoV-2 is infecting white-tailed deer. It's doing so within the U.S., but this is a different study out of Canada that has some interesting implications, so that I thought we could do it. And one of the things that I think is useful.
And also the timing of us talking about it is fortuitous. If anybody out there is a hunter, we're in probably deer season, depending on where you live. I think Massachusetts shotgun season starts up soon.
We are firmly in deer season here.
Yes.
Dixon and I were talking before we started recording, and I mean, we have tons of deer on our property. Every night, a dozen of them walk through, and they always, in the spring, a few of them have babies, and my wife always gives them apples. And, you know, if she has some SARS-CoV-2 on her hands, maybe that's one way of infecting them, because it's not a lot of food for them. So he throws that.
Because the other deer ate all of it.
Yeah,
is the problem.
Well, remember, we got rid of the predators, and that's
yes, we got rid of the wolves. The reason we have this problem is that we got rid of the wolves and cougars, and now we're stuck with the deer, and we are not doing as good a job of controlling them as the wolves and the cougars used to do.
No, we are not.
This summer, I was in the garden, and I left the gate open, and I turned around, and the deer is in the garden eating the spinach, you know. I'm a few feet away. They don't
just
care.
Brazen, yeah.
They just want it, and they're always around.
Suburban deer are not. They're not afraid of humans because they're not routinely hunted in those areas.
Yeah, so. That's right. I don't hunt them. I I say, just chill, we'll get you some apples. But lately, the males are in heat, right? Or whatever. I don't know who's in heat. The males are chasing the females.
It's called the rut.
The rut. The males, there's one male with a lot of points, and there's 6, 7 females, and they're always, he's chasing them.
Points is good.
It's Vincent's deer knowledge. This is great.
What's wrong with points?
Points is good.
Points is good, yes.
I don't have any deer knowledge, I admit, but I did drag a few from the lawn into the woods before. Anyway, at this time of year, the single male is chasing all the others. But you know what's funny? So sometimes, we're giving them apples, and there are 6 or 7 females, and they're eating the apples, and then a male comes and tries to eat an apple, and they chase him away.
Oh, yeah.
He's totally afraid of them, except when he wants to mate with them, right? Then he chases them. But
right.
in the pre-mating season, no apples for you, buddy. It's really funny.
And there is one other behavior that's kind of interesting, and that is that if you're driving and you see a deer and you stop, the females always go across the road first.
Yeah. And
then about maybe 2 minutes later, the male comes out. So the females are like, you can take a hit from a female, okay? And by the way, the cougar is also a car, so perhaps the predators are not all gone, but bad analogy. The males use the females as their shield. And if they've got a harem, they stay last. And the same is true for all the other hoofed animals. Like I was in Africa not too long ago, and all of the antelope and hoofed animals over there, they behave similarly.
I don't think it's so much the males using the females as a shield as it is the females have an agenda which is very practical. They're hunting out food. Yeah, and the males are following the females.
Okay.
Anyway.
The females cross the road first because they're on their way to acorns or whatever, and then the male is following the females because that's what's on his mind.
So they're living near us, and there's ample opportunity for them to get infected, I guess, from the apples. I don't know how else they would be infected by us because we don't really have contact with them. But
people put out feeders for them. And,
I suppose.
They come in contact with peri-domestic animals and domestic animals routinely.
Who knows? Anyway, so it turns out that SARS-CoV-2 can infect at least 54 non-human mammal species. It's a lot. And that includes some where reverse transmission has occurred. They call this zooanthroponosis or reverse zoonotic disease transmission. In other words, the non-human animal gives the virus back to people. That's been shown among farmed mink in the Netherlands, which we talked about a while ago. And I love the name for mink, Neovision Vision. Isn't that cool?
Really? Is that the name?
Neovision.
That's good.
Zooanthroponosis, I thought that was people giving it to animals.
No, it's animals giving it to people, right? Zoonosis is animals to people.
Correct.
The first time.
Yeah.
The first time.
Zooanthropo is back. Yeah.
Right.
And hamster
Anthronosis would be the intermediate, us giving it to them.
The colloquial term for that is ping pong Charlies. It just keeps bouncing back and forth between animals and humans.
Yeah, and we've talked about the deer situation with SARS-CoV-2 before because people have found it and found there have been serosurveys as well. And it seems to be, just from the observational surveillance studies that have been done so far, it seems to be pretty widespread in North American deer whenever people look. And it seems like it's probably spreading deer to deer, which makes sense because they're very social and they lick each other's faces and that kind of thing, and that would spread it. So this paper kind of takes things a step further, and they do a survey and really kind of track what's going on with this virus and also sequence it.
So besides the mink spill back into people, we have also from hamsters, pet hamsters. I think in Hong Kong they infected people. So they culled thousands of animals, mink and hamsters, to stop these spillbacks. All right, so the white-tailed deer is Odocoileus virginianus, a common North American ungulate. The deer develop subclinical infection, And previously, deer have been sampled in Michigan, Illinois, New York, Pennsylvania. 40% of free-ranging deer positive for SARS-CoV-2. Transmission, as Ellen said, amongst deer. Multiple spillovers from humans to deer have often been reported. And there's some evidence for sustained transmission in deer. There's a divergent alpha-like virus that's been reported in deer. And so they say in the introduction, virus isolates circulating in animals may become less fit for humans, but some changes may occur which have consequences for people. So you really need to know what's going on. And that's
and it's not even fitness for humans. It's just that's giving more opportunity for the virus to replicate and more opportunity for the virus to get mutations that, could do all sorts of things.
Exactly, right. Which might be different from what's happening in people because it's a different species, right?
Right.
That's right.
So this study was done from November to December 2021.
Last deer season.
The other night, I thought that we were in 2023 already.
Sometimes it feels that way. I agree with you.
300 white-tailed deer sampled from southwestern and eastern Ontario during the hunting season. Most of them were adults.
And so the way they did this, they already had a program. When hunters get a deer, they usually have to check in somehow to report that they got it. And this is often done. It varies from place to place, but it's often done at a check station where you bring the deer and they verify. Oh yeah, that's a buck. You used your tag. And in the process, they'll piggyback on these various types of studies. And so they've been doing a chronic wasting disease monitoring study, testing for that in the deer that people bring in. And it's a very convenient sample to work with and also is relevant epidemiologically because these are deer that people are, gutting and working with, and you kind of worry about transmission in that setting. And so they just went with that, and they're taking swabs. In addition to the CWD screening, they're taking swabs now for SARS-CoV-2.
Well, Alan, you just raised an interesting point, though, because I've obviously been out at this time of the year to go fishing, and you have to be very cautious when you go fishing during the hunting season.
Or hiking.
You should wear something that
wear some blaze
tells
orange.
you are not a deer. I have a big T-shirt that says I am not a deer. So, and it's spelled correctly too.
No, better not to have the word deer on the shirt at all.
Yeah, but the thing that I'm curious about is, have they ever done any studies on the places where they dress them out? Because there are local, right next to the checking stations, there are usually, you can have your deer dressed out, butchered, packaged up, and frozen, and now you can access it anytime you want. You don't have to do anything else to it except kill it.
Yeah, that was kind of my thought, is that I think that those are paired, as you mentioned. So I think that the check-in station and the places where you have the dressing done are usually linked together.
They're frequently they are. Some places it's more of a local butcher shop sets up somewhere down the road. But
But what about those people that are handling hundreds of deer?
That's a major occupational exposure, certainly for chronic wasting disease and potentially for other things as well.
Exactly.
So listen to Richard Knight talking about prion diseases. Yeah, I said at the end, what's the danger with deer and CWD? He said, well, we don't have it here in the UK, but people buy deer urine.
Ooh.
Because setting hunting traps or something?
Yeah, for bait.
Right.
So just like you were talking about earlier with the males following the females, you could get some female deer urine to try to entice a male deer to come if you were going to hunt him.
That's actually, I think you can mail order doe in estrus urine.
Exactly.
And there's like a whole line of products that are designed around this that you can hang up or spray around or whatever to
Alan,
lure
wouldn't
deer.
that be a female order rather than a male order?
Yes, it would be.
Well, look at this knowledge about deer on This Week in Virology. This is just great. And we'll get people writing in, I'm sure. Anyway, Richard said that the deer urine is imported and could have prions in it as well. Anyway, from this study, 213 nasal swabs and tissue from retropharyngeal, behind the pharynx, lymph nodes. Obviously, these are dead deer.
Yep.
They did PCR, RT-PCR, to look for SARS-CoV-2. So 5 of the 213 nasal swabs were positive. 16 lymph nodes were also positive. So overall, 21 samples were positive. That's like 6% of hunter-harvested white-tailed deer, all from southwestern Ontario, most of them female, 65%.
At this point in the paper, they talk about the CT values as being less than 40 and less than 36. And
Yeah.
I was less than impressed. But then later, they actually mentioned CT values of around 15, 16, and so forth. So that was more convincing for the actual positive ones.
Yeah.
Right. So, Vincent, usually there's a season for male and female deer, and they're separate because they want to harvest female deer. They value the males.
It varies from state to state and within a state.
Yeah, no, you're right.
My part of Massachusetts, Western Massachusetts, we don't have much of a deer problem because we don't have so many of the habitats that they like out here, ironically, on the rural part of the state, because a lot of the farms have stopped being farms and now it's hardwood forest. The eastern part of the state, where you have these sprawling suburbs going all over the place that are de facto wildlife preserves, are absolutely infested with deer.
This is true.
And Martha's Vineyard and Nantucket, which are islands where the deer are not native, the deer are completely out of control.
Yeah.
So they in well-run programs, a state will have regions where they'll set up, say, okay, kill all the does you want over in this area, and then they'll have more stringent, or different parameters someplace else. But it varies. There'll be seasons for these, separate seasons sometimes, or there'll be, you can kill this many of one or this many of the other, that kind of thing.
Alan, how do you know so much about deer? I have to ask.
I've read a couple of books about this, and I have relatives who hunt, who are really on top of this stuff, and I got interested in it. It's like, hey, wow, this is a really deep subject. In fact, I think I had a pick of the week a few years ago that was a book that was just like an overview of the deer situation across the U.S. and how it got to be the way it is, and it's fascinating stuff.
How many deer live in the U.S., Alan?
Forget about it.
Hundreds of millions, I think.
Hundreds of millions.
I
don't know. It's at least tens of millions, but it's a lot. It's a
It's
ridiculous.
quantitative. A lot.
We have a ridiculous number.
It's estimated there are more deer living in New York, in the United States today, than there were when Columbus landed.
Yes.
Are there more deer than people? That's what I want to know.
I don't know how the figures compare.
Can we? Let's just look it up. Let's see how many deer
million people
live in the U.S. 36 million.
36 million. Okay.
Well, so that's
tens of millions.
10% of the human population.
And
yes,
I'm sure it's not equal in every state, right?
No, it varies a lot.
All right, back to the virus.
Yes.
Virology.
Right,
this is the way TWiV is supposed to be. We're supposed to diverge into what interests us and make a lot of mistakes.
Talking about the virology. Come on, this is all
so
part of this.
They did some sequencing, which shows that you want to do phylogenetic analysis, and it shows that these genomes are a diverged clade within a certain lineage, the B.1 Pango lineage or 20C, two different ways of naming the lineages. They share a recent most recent common ancestor between May and July 2021. It's the backbone that they're derived from which the beta, the epsilon, and the iota variants and some mink variant outbreaks emerged. And the closest genomes in the database are human-derived sequences from Michigan, sampled about a year before. Michigan!
Okay, so now I have to tell you what deer information I had to look up.
Okay.
Because I looked immediately at the map in Figure 1, and I said, how well do deer get across bodies of water? Because between Lake St. Clair and Lake Huron, there's a river, and between Lake St. Clair and Lake Erie, there's a river, and they swim. They are really good swimmers.
They are.
So no question that the deer could be going back and forth. They are not paying attention
People
to...
have actually been boating out on the Great Lakes out of sight of land and have come across deer
That's
swimming.
Right. That's right. That's exactly
Just
right. But
so
deer...
I can't remember when when Canada really opened up to the United States with respect to closed borders because of the pandemic, but it didn't matter because the deer were going
They were
back
not
and
checking
forth.
the passports on the deer.
Right.
No. But at 3:30 in the morning, Kathy, it's probably not too crowded on most bridges that go across those rivers. And deer take advantage just like coyotes do. I mean, up until a certain year, there were none east of the Mississippi, coyotes, that is. And then all of a sudden, coyotes appeared. So some pregnant female coyote calmly walked across one of the very long bridges across the Mississippi River and started a whole new population on this side. And now look what happened. You can't even take your dog out for a walk anymore. So it's really amazing how quickly nature fills the gaps. And that's what's happening here. That's really...
speaking of swimming, when I was in graduate school, I used to go to Plum Island, an island off the tip of Long Island, with a substantial body of water between the mainland. It's called Plum Gut, and it has on it a high containment animal pathogen facility.
Indeed, it does.
And they're very careful. I remember talking to one of the scientists. They're very careful not to release any viruses on the island. I said, well, what does it matter? There's nothing here. He said, no, the deer routinely swim across the gut. He said, you can see them. They're just their head is above the water. Yep. And they're moving, and they get to the island, they hang out, and then they go back. So they could conceivably bring viruses, including foot and mouth disease virus, right back and forth. So they're very careful. Anybody from Plum Island listening, hopefully you will.
Well, does anybody live on Plum Island?
No, nobody lives on it, but people work on it.
Oh, you mean the people who work there, yes.
Yeah, I know a number of virologists who work there. And the idea is actually to close it down and move it to Manhattan, Kansas. I don't know if that's still happening, but they built a new facility.
They wanted someplace more remote.
They built a new facility, a BSL 3-4 facility in Manhattan, Kansas, and the idea would be to shut this down because you have to take a ferry to get to the lab. Exactly. Every day. I did that a few times as a graduate student. It's quite an interesting experience. You have to take the ferry, then you go in, you go to a locker, you disrobe, you put white clothes on that are very starchy and itchy, and you go work. And then you come out, take them off, you shower, and then you get your clothes on. And the funny thing is you go in and you meet people for the first time naked. Peter Palese said to me when we were going the first time, he said, Vincent, it's very strange. You're going to have to look up when you're shaking people's hands. And it was true. It was such a weird experience because you go in and everybody's got, all the men are separate from the women, of course, but you meet people and, oh, hello, Vincent, how are you? That's Plum Island. And the deer are deer that go back and forth. So this lineage, the Ontario white-tailed deer lineage, gets its own name, B.1.641. Okay?
I think this should be called venison pie.
That's good. Venison pie is good. Anyway, so they think, they say there's a little bit of uncertainty in the placement, but for a number of reasons, the proximity of Michigan to southwestern Ontario, as Kathy pointed out, mix of human and animal cases say they think this is probably right, this placement of the viruses there. So, and they also looked at recombination. There's no indication that recombination gave rise to this lineage.
And the phylogeny of this, I mean, this thing is just out on its own limb.
It is.
Look at, if this is an open access paper, by the way, and you look at the the phylogenetic tree they've got, and it's, the clusters of various things in various species, and then there's just this line out to this B1 641 in the deer. So it came over to them from somewhere and has probably been in the deer for a while.
Yeah, the other thing that's so interesting about that figure is that if you look at the phylogeny, you see different groupings. They're all different colors, indicating the virus that's coming from different organisms. That's been sampled from different organisms. And so the idea that, the virus is perhaps adapting a little bit to different organisms could be something you see. But what's also very cool is that the B.1.641 is sort of comes out of a whole bunch of green dot viruses, or is placed within a clade within a bunch of green dot viruses, all of which are from neovision or the mink. And so thinking about ways that these viruses might be spreading from animal to animal to animal before potentially ending up in humans is also an interesting thing to think about.
They also have a human-derived sequence from Ontario, which was very similar to the deer. That 80 out of 90 shared mutations, and forms a separate group within the white-tailed deer samples.
Right, and so one of the other things that was going on at the same time in 2021 was Ontario had a program to sequence 100% of the COVID-19 test samples that they got, if they could get sequence out of it. So they had this trove of data there, and this one human case popped up in there.
Yeah. They say this has, there's evidence that this human case, the virus from the human case, belongs to the B.1.641 clade, and it also has epidemiologic link. Apparently, well, first of all, it was collected in the same geographic region, southwestern Ontario, so the human and the deer cases. And the human case had known close contact with deer the week before symptom onset. No known contact with any individuals who tested positive for SARS-CoV-2. Although, that's iffy. You just don't know, right?
Yeah,
right.
It's asymptomatic. So this human isolate is so close that you have to blow this figure way, way up till you can see the yellow human dot under the red deer dot for this lineage.
Yeah. The deer dot. It's good.
The deer dot.
And then they look at the kinds of amino acid changes in the genomes, and these are deer specific changes. A lot of them have been seen before in animal derived sequences, and they're trying to understand whether there's been any selection pressure on this B1.1.641 lineage. And I won't go into the types of analyses they do, but they conclude that these viruses have been probably transmitting among deer for a while with minimal immune selection pressure, because the genome changes are not consistent with immune selection. So, I mean, don't deer make an immune response, Brianne?
They are jawed vertebrates, so they have they've got an adaptive immune system. But of course, as you were starting to talk about the immune evasion, I started thinking, what do we know about deer immune systems?
Not much.
And there certainly are other unique aspects of some other species that I can think of. I can think of some unique things in the cow immune system or in the llama immune system. So perhaps there are unique things in the deer immune system as well that are not well studied.
So basically, there's no evidence for changes in the genome that might be a consequence of immune selection, all right?
Well, another possibility is that the deer just haven't gotten widespread enough infections to have applied that much pressure. Early in the human pandemic, we didn't really have selective pressure,
correct,
because there were plenty of hosts around. So there was no. It's when it gets to be a problem for the virus to find the next host that you get that selective pressure. Okay. So it may just be a numbers game.
Yeah, I like that. Good point. Yep,
that's a good point.
All right, so then they tried to isolate some viruses. Very nice. They had to put some of their samples on cells, and they get cytopathic effect. And they get virus. So what can you do with virus from deer?
Plaque assays.
You could do plaque assays, and you can say, is it neutralized by sera from either vaccinated or convalescent humans, right? So, I mean, that, remember Michael Letko has this Costa spike from Russia that apparently is not neutralized by convalescent and vaccine sera. So what's going on here? So they did neutralization assays, and then all these isolates are recognized to a similar extent as the ancestral SARS-CoV-2. And they used Omicron as a control, which is not recognized well, which is nice. So they conclude that these changes in the spike in the white-tailed deer isolates have no impact on antigenicity, which is good.
Well, so basically what this says is that it has no impact on antibody antigenicity compared to vaccine-derived serum. So as a person who is vaccinated with BNT 162b2, I do not have to fear if I go hang out with deer that I will either infect them or acquire virus from them.
You do not have to fear the deer.
You got any deer in your backyard at the moment, Brianne?
At this exact moment, no, but today, yes.
Yeah, okay. Do you ever go out and commune with them?
No.
Not usually.
Okay. I mean, I go out and they're standing there, 5 feet away, and I often wonder whether they're going to charge me. I talk to them. I would say, what's happening? You want some food? Just wait. I'll go get some. And when I go back in and get some and come out with some apples, they're gone. They don't wait. They're not patient. Okay. Anyway, so that's the story. The deer are infected. The viruses aren't too much of a concern so far to people, but we don't know how this happened, right? From humans to deer. They say maybe there's a intermediate host, as we mentioned, mink or some other species. They have there's an interesting figure here with some different scenarios for how this virus ended up in deer. And they say spillover in deer followed by deer to human transmission is most likely scenario. So humans to deer, spreading in deer, and then back to humans.
And they have these big stretches of what they call unsampled viral evolution in the graph, meaning we don't know what the virus was doing here. We just don't have the data. It is interesting to me that they drew the graph as all the scenarios go from Michigan to Ontario and not the other way around.
Yeah, well,
Yeah.
Canadian authors.
Yeah. Every time I see things like this, I remember an earlier episode we had with Tony Shoutz where he was talking about SARS-CoV-2 viruses in mice.
Yeah.
And wild mice. And I wonder how wild mice could be part of this.
Yeah.
The other thing that's interesting to me is that the lineage that's shown in the main body of the paper doesn't have years along the bottom. But if you go to the extended data, there's one that does have the years. And that allowed me to confirm their supposition that this lineage diverged sometime in 2020. And if you look at it, there's this long stretch of time where who knows what it was doing in the deer, but evolving away from where it came. And that length of arm is longer than it depends on how you look at it, but as long as or longer than the Omicron arm. So to me, it was interesting that because we think of Omicron as having, evolved somewhere or somehow without much sampling, and then suddenly there it was. And this is kind of the same way.
We haven't really discussed much about deer farming, but there is that to discuss because there are huge herds of domesticated deer that are used for sources of meat. And it's possible that the wild deer, right, maybe the farmed deer caught it first and then gave it to the wild deer that sort of come up to the fence and say, what the hell are you doing in here? Why don't you jump over it?
Or the farmed deer that escaped.
Yep, all
all
of the above.
Oh, interesting.
I don't know if there is mink farming in Michigan or Ontario, but the proximity of the mink and deer lineages in this is kind of interesting.
They even farm elk out west. So
Has anyone sampled farmed deer? Do you know? Does anyone know?
Right, I'd like to know that, actually.
Not that I've seen.
Where was that in one of those
I thought.
papers from a year ago?
I thought it might be, yeah.
One of those earlier papers?
Let's see. Let's check it out.
It does look like there's mink farming in Michigan.
There is, okay.
And in Ontario.
So that's another plausible sequence of introductions.
Yeah, exactly.
Human to mink.
Yeah, in late September 2020.
And there's a lot of escapees in mink farms. Minks are very clever animals. They can chew
weasel
their way
their
through
way
a lot
out.
of stuff. That's right.
I just found a Nature article from April of this year. It says COVID is spreading in deer, and they have a picture of a guy swabbing a deer, and the deer is like, arr, arr. He's got its mouth trying to move away. Yeah, so it's a live deer, and he or she is trying to swab it.
That's
it's very
remarkable.
interesting. It's in Texas. They don't say anything about sampling farm deer.
Wow.
I don't know, but it's interesting. Anyway, so the last point they make is that, we're not really doing a lot of wildlife sampling anymore in deer or other mammals. So really don't know the extent of infection that's out there. And it really would be wise to do more, right?
Yes.
Yes.
It's a great idea.
To see who's infected, what kind of genomes are evolving, whether they pose any risk. I mean, you do some neutralization assays, you could figure that out. You don't even need virus. You could just put the spike gene in a pseudotype. So maybe we need to pay more attention to this, right? I haven't seen, for example, many papers on other wildlife.
Yeah.
Wildlife,
No, not at all.
animals out there, not in the lab, right? Anyway, I thought that was cool. Now we have another paper which is even cooler. If you said,
Well, I don't know about that.
If that's
but
possible.
This is pretty cool.
This paper, I don't know. This second paper, I was pretty impressed by the whole
it's
idea.
amazing. If you had said,
Yeah.
could this happen? I said, nah.
Exactly.
No, not happening. But
And the
yeah.
figures, which we'll get to, are pretty trippy.
trippy. Is that like far out, Alan?
Yeah. When I was looking at that, I was like, wait, what's going on here? And I read the legend, and I read the legend again. I said, seriously?
And,
Yeah.
and if you think about it, the experiment is not that conceptually difficult.
No.
Yeah, I
Or
mean,
technically difficult.
in fact, I was sitting here thinking about all of the different types of permutations that one could think about for this experiment, and I'm like, those seem pretty straightforward. I wonder if anyone has actually done those.
Yeah, sure.
So we
this
need
is
to introduce the paper here.
Nature Microbiology. Co-infection by influenza A virus and respiratory syncytial virus produces hybrid virus particles. 2 viruses in the news, right? Currently circulating. Authors are Hani Vijay Krishnan, Streetley, D. Goldfarb, Clark, Mullen, Carter, Bella, and Mercia. Where are they from?
All from University of Glasgow.
And David Bella is a structural guy. He had sent me this a couple of weeks ago, and he said, this is pretty interesting. You might want to talk about it on TWiV. So thank you for that, David. This is all about co-infections, right? People can be co-infected. As Daniel Griffin always says, there's more than one virus out there, and they can co-infect you.
But this also gets at co-infection not of just an organism, but of an individual cell.
A cell, yes. They're starting with a cell, because the question of what happens in people is very interesting, but not addressed here. But they say something in the beginning. It says co-infections by more than one virus represent between 10 to 30% of all respiratory virus infections and are common among children. Kids get infected with more than one virus. Not surprising, right?
Yeah.
And so
They've got a lot of viruses to get through in a few years, and you got to stack them up.
What are the clinical implications of co-infections? We don't know. Sometimes there's no implication, there's no effect. Maybe sometimes the disease changes. I think we did a paper a long time ago from Glenn Rawle, which said that co-infect mice with 2 viruses, you get a very different disease than either virus alone. But those are mice, of course.
Well, and there's not all co-infections necessarily work, because I know we've talked about papers where there's exclusion. So if you get one virus in first, it prevents infection with the other in that cell.
Yeah. I mean, so I'd like to know why they did this, actually. So if anyone's listening, obviously they say, well, you know, right now flu, influenza virus, and RSV are co-circulating. So we decided to do a co-infection. Is that really?
That's not. No, I'm imagining like trying pairwise combinations of viruses, just see what co-infects.
No, this is how old is this paper? When was it first?
November. It just came out.
Yeah, but how long did it take for them to do the research? That's about a year old, right?
Received August 2021.
So maybe RSV and influenza wasn't so prevalent then.
Oh, this predates the current season for sure.
Exactly.
So they were.
No, I agree. I don't know. I think that if I was thinking about the epidemiological data, and thinking about co-infection, I'd think about, is there the pathology in the respiratory tract or the cytokines in the respiratory tract following one infection influencing what happens with the other. I don't know that my brain would immediately go to the same cell being infected by two viruses. I would more be thinking about the same tissue or something like that.
Yeah.
So how they got to the same cell is really interesting.
Also, what struck me is that, yes, they're both negative strand RNA viruses, but one replicates in the nucleus, one replicates in the cytoplasm.
Yeah, that's right. So let's talk about the two viruses, influenza virus, respiratory syncytial virus, both negative strand viruses. The influenza genome is segmented. The RSV is not. As Kathy said, nuclear reproduction for influenza virus. The glycoproteins are different, right? Influenza virus has a hemagglutinin and a neuraminidase, and respiratory syncytial virus has HN, hemagglutinin, neuraminidase, and F for fusion, so different glycoproteins. Although I guess they're both released from cells by budding, right? So that's a common assembly pathway. They have nucleoproteins coding the negative stranded RNA. Okay, so they do co-infections. What are the cells that they're initially using? I can't remember.
A549.
A549. So a human lung cancer cell line. And they do high multiplicity of infection so that every cell will get both viruses, and they look at replication. They do growth curves basically in singly or co-infected cells, and they say, you know, influenza virus is not really impacted by co-infection, but respiratory syncytial virus is. You reduce the titers like over 400-fold in the presence of influenza virus. Okay,
and they try to muck around with the MOIs to even them out, but it stays. Flu keeps pulling ahead, basically.
Right. Okay, then they say, where are the viral proteins in co-infected cells? So they either single or co-infect cells. They fix the cells and then do immunofluorescence microscopy with antibodies to viral proteins. And so, of course, in the singly infected cells, the virus just have the features that we know of, like the nucleoprotein in RSV is their inclusion bodies. It's found in inclusion bodies. For influenza virus, it's diffuse. We know that before. And they find in co-infected cells both. So RSV is punctate and influenza virus is diffuse. So it's not affecting the localization. These viruses both bud from cells in lipid rafts, which are just specialized parts of the microdomains of the plasma membrane.
Can I just pause for a second?
Yeah.
Dixon's video is freezing. No, Dixon is freezing. I'm sorry.
Dixon, are you cold? Mind. Are you cold, Dixon?
No. Good.
No, it was just in one position for you were holding very still.
I'm very thoughtful.
Yes.
You know, this gives me the opportunity to mention that the last time you were on TWiV, Dixon, you had a power failure, remember?
I did.
When you came back, you were actually not recording, so the rest of the episode had no Dixon.
Oh, dear. I thought you said...
Your letter reading was gone, your pick, so I had to edit all that out because there was no Dixon. So it was a much shorter episode, Dixon.
I can imagine.
Anyway, so your picks from last time...
I'll try to be more...
Dixon,
yes.
So this is to remind you, your picks. You should do your pick from last time because that was never recorded.
I will.
Okay. Anyway, back to lipid rafts. So they stain for the glycoproteins with antibody for both of these, right? They go hemagglutinin for flu, the fusion protein for RSV. The HA is diffusely around the plasma membrane in both single infected cells and co-infections. RSV-F is localized to regions containing filamentous structures extending from the cell surface. Those are the budding virus particles. And in co-infected cells, the HA was not excluded from these RSV budding sites. Very interesting. So the HA is still being produced, and it's in there amongst the RSV proteins, F protein. All right. So
I mean, that kind of makes sense because the plasma membrane is having things diffuse randomly throughout. And so you shouldn't expect that there's going to be total exclusion.
I don't know. You could imagine that there might be some exclusion by certain glycoproteins, but yeah, it doesn't happen. But so there's mixing, and they say, okay, maybe you could get virus particles with both glycoproteins, right? That would be the next step. So they used super resolution confocal microscopy. Super resolution confocal to look at virus particles to which they've added antibodies for the HA and the F protein. And they saw dual fluorescent particles, so suggests that particle has both glycoproteins, right? But interestingly, the glycoproteins are in distinct regions along these filaments of budding virus positive. So the influenza HA is at the distal end of this filament and not just throughout the whole filament of these particles. They also saw dual fluorescent particles distant from co-infecting cells, which they say suggests that, we're getting virus particles budding off and we're still getting these dual glycoproteins on them. And then they also saw individual influenza and RSV virions, right? So they see these hybrids and they see some individual ones as well.
Which is kind of what you'd expect.
Yes.
If you infect with both of them, you'd expect some cells to just get one virus or the other and replicate that.
But what's weird here or interesting is that we seem to be getting some hybrid particles, right? So we're going to look at those some more. They do microscopy on live cells now instead of fixing them to try and get more information on what's going on. They see these bundles of filaments coming off the cell surface. They have lovely pictures of that. F is along the length of the filament, and at the very tip is the HA. Very interesting. And they use scanning electron microscopy to further look at that. All kinds of imaging in this paper, right?
It's really beautiful. And
it's gorgeous.
They also do everything really nicely color-coded in all their figure legends and figures that make it easy to follow.
Yes.
So they have...
figure 3 was the one that had me kind of staring at it for a while.
Yes.
Like, whoa.
Yeah, that's beautiful. That long... In fact, that's what they see from this live imaging, these long filaments with the F along the whole filament. The HA is at the tips.
Yeah.
Do you have a way to share in this program?
No, I'm sorry, there isn't.
Oh.
We might switch to another...
but so I think, and this isn't open access, is that right?
It is.
Oh, it
yes,
is.
anybody can download this paper.
Yeah.
Okay.
They can.
Because figure 3 is going to be, I think, an iconic, the part B where it's colored in.
Yes.
I in the margin I wrote, this reminds me of the Fred Murphy famous Ebola image.
Yes.
This is the one that the popular press should pick up.
If I didn't know what I was looking at and I looked at F, I would think that is an enlargement taken from the International Space Station of an incredibly jammed suburban region with loops of dead ends and lots of houses, and I can see some farms, and they said, and so-and-so lives in the blue and such lives in that sort of thing. So there's a lot of pattern repeats that nature uses, and we are part of nature, so we do that. It's remarkable, though, that you can get this resolution. It really is quite remarkable.
They move on to cryo-electron tomography now to look at the structures of these particles, and they say they look like hybrid viral particles with elements of both viruses. And they have two kinds of particles that they see. True hybrids that they call HVPs, hybrid viral particles, and pseudotyped viruses, which would be like one virus with the glycoprotein of the other, which we always talk about as, SARS-CoV-2 pseudotypes with the SARS-CoV-2 glycoprotein in a lentivirus or in VSV. We make those in the lab, but apparently you can have them generated in culture. So the hybrid virus particles have structural regions that are like influenza virus and RSV, distinct structural regions. You can see the ribonucleoproteins in their specific parts of these filaments. You can see the glycoproteins either at the ends or at the body of the particle, and they can see the matrix proteins. They also find some virus particles that look like these RSV. So the RSV forms the filaments, and they have a different glycoprotein ordering than they would have expected. They differ in shape compared to RSV alone. They say they have a triangular arrangement, which is more like the influenza virus glycoprotein rather than the kind of density you would see with RSV glycoproteins. And they say these are the pseudotypes. So probably you have a different matrix underneath it, which is influencing the way the glycoproteins are arranged. Let's see. So basically, co-infections give you two kinds of hybrids that are distinct from either parental virus. And the picture's worth 1,000 words. You can see. Looks beautiful.
Yeah. You have to go and look at this paper and look at figure 3A and 3B online.
Yeah. I can tell you that 3B is going to be the image for this episode for sure.
Yeah.
Cool.
It's so beautiful.
Yeah.
But you have to go look at it. So what about the antigenicity? This is very interesting now. I mean, the whole thing is interesting, but now it's very cool. Do these particles have altered antigenicity? So how would you do that? You just do neutralization assays, right? With antibodies, you have HA antibodies, you have RSV antibodies. So let's start with anti-influenza virus HA antibodies. They get viruses from infected cells, co-infected cells. They are not as well neutralized by anti-HA antibodies as viruses collected from a single influenza virus infected cell. So in other words, the hybrid virus is not being neutralized as well by anti-HA antibodies. So there's clearly something different about it. And then for RSV, they have a monoclonal antibody, palivizumab, which is clinically used to treat people with RSV infection. And so they try it's a neutralizing antibody. They try neutralizing what you get from single and mixed infections. So this is really well neutralized by the palivizumab, in contrast to the anti-HA antibody. And so what they conclude from this is that the RSV cannot enter cells via influenza virus glycoproteins because the anti-RSV antibody neutralizes it really well. It doesn't seem to be using influenza virus glycoproteins. Otherwise, you would have reduced neutralization, right? I think that's cool.
Yeah.
So despite the fact that these are hybrid particles, all is not equal, right? It's like both HA and F are not doing what they usually do in normal infections. Okay, the next part, another bunch of cool experiments. Is the receptor tropism of these viruses different? Because now you have two different receptor binding glycoproteins on the particle, so what's the consequence of that? So what they do is they treat cells with neuraminidase, which will remove sialic acid, which is the receptor for influenza viruses. Okay, so the control is this blocks entry of influenza virus into cells using influenza viruses by itself, right? If you use virus from a mixed infection, influenza virus entry is increased after treatment of cells with neuraminidase. It's more. And for RSV, there's no difference because RSV entry is not dependent on sialic acid. So the increase...
So flu can use RSV's code to get in, but
RSV can't use flu's code.
True.
That's right. So they say the increase in influenza entry into sialic acid deficient cells in hybrid particles indicates that these have expanded receptor tropism. So then they want to know, all right, is this due to the glycoprotein? So then they can use their antibody to F, and they can see a reduction in entry of influenza virus in neuraminidase-treated cells where the entry would go up. In the presence of pavlizumab, it goes back down again. So those viruses are using the F protein to get into cells. Population of influenza virus dependent, hybrid particles dependent on RSV-F. So they think that that's actually the functional glycoprotein of these particles, the F, not the HA of influenza virus. It seems to be driving all the entry. What about the genome? So they do some experiments that suggest that the genome of both viruses are delivered into the cell. Remember, these are hybrid virus particles. They have influenza and RSV genomes in them. They do an infection. They can see both influenza viral genomes and RSV viral genomes. And this is apparently stable over multiple passages.
And let's just make sure I have this right. The way that they know that is from these EM pictures.
Yeah.
And they're just basically looking at the structure and saying, this is the structure of the RSV genome and this is the structure of the influenza genome. And they're very characteristic patterns, so they can say that. But it's not like they're doing some kind of nucleic acid probe or anything like that.
No, that's right.
Yeah, this was sort of one thing that confused me a little bit, or I don't know if confused, but it made me think about some things a little bit, is that back at the beginning, when they talk about co-localization and some of these hybrid particles, they talk about, 56% of cells displayed these dual positive filaments in the co-infected cells. So I guess my understanding would be that the co-infected cell is making some hybrid particles, some influenza, some actual normal influenza, some actual normal RSV. And in all of these subsequent experiments, they're using just sort of the virus soup, virus-containing soup from those co-infected cells. They're not actually sorting out and looking at what just the hybrid particles do or what the pseudotyped versus the hybrid particles do. As someone who does not do that kind of work, I can realize that would be really hard to try to sort out these particles, and I understand why that is. But I sort of wonder how that impacts on a lot of what they're looking at here.
Yeah. It's a good question, I think. Yeah. So anyway, the final observation is that, and I'm not going to tell you how they did it because we're getting too deep into the weeds now and you're probably already lost, but these hybrid particles, remember, they're dependent on the RSV-F to infect cell. They can lead to the spread of influenza virus from cell to cell. They do passaging under an overlay that prevents diffusion. And they can see the influenza genome being passed from infection to infection of these hybrid virus particles. So the hybrid virus particles can mediate the spread of influenza virus from cell to cell.
I think getting at Brianne's question, this is where I wrote in the margins when they were describing kind of what experiment they were going to do next. I wrote, how can they rule out that single particles aren't being produced in their mixed infection that are going to complicate this?
Yeah.
And I think it's because they do this experiment with the treatment with the neuraminidase.
That's correct. They do.
So that specifically rules out anything that requires the neuraminidase.
That's right. They use neuraminidase, and still the influenza genome is able to go from cell to cell in those conditions. So it's using the RSV glycoprotein. Yeah. right, so all this is done in A549. The final thing they do is say, let's do primary human bronchial epithelial cells. And it's really a subset of all the experiments they've done, but they do see hybrid viral particles being produced in those as well. So this is very interesting. You're getting hybrid viral particles. And they have properties of both viruses. I wonder what that means. Well, first of all, do you see it in people? They say we have to look in people. They have the potential to be generated in vivo, but I guess you'd have to look, right?
And wouldn't now be the time to try and get those samples?
Yeah, I would guess, yeah. I mean, you kind of, it's not an easy experiment, right? Because you have to get a lot of samples and make sure two viruses are present in the same person, right? And then you have to look to see if you get these hybrid viral particles with glycoproteins from both viruses. Yeah. I'm sure they're doing it, right?
It
or
would
trying
certainly
to.
give an interesting way for influenza to evade antibody.
For sure. Yes. Especially since we don't have any vaccines against RSV, right?
Right.
And potentially reach new areas of the lung.
So that's a good point.
Doesn't
Yes.
RSV get lower sooner than flu usually does?
In fact, they mentioned that, flu predominantly infects the upper and middle tract, RSV more readily spreading to the middle and lower. So this could change the kind of disease you get, right?
Right.
So I think this is important. I think this warrants further investigation for sure, right? Very good stuff.
Yeah, when I heard about the paper and saw the title, and I thought, okay, extraordinary claims are going to need extraordinary data to back it up.
Okie dokie. Time for some email. Let me take this first one.
Good.
So I shouldn't be laughing, but this is with respect to TWiV 896, which was some time ago. And this was an episode where it was me, Dixon, and Amy. And we did a paper from Rockefeller, which the three of the authors, the senior authors, have written: Theodora Hatzianu, Paul Binash, and Michel Nussenzweig. And they wrote, to the host of TWiV, we listened to your commentary of our paper, Increased Memory B-Cell Potency and Breath After a SARS-CoV-2 mRNA Boost in TWiV, episode 896. We were embarrassed for you by the way you misinterpreted the contents of the paper and by the numerous factual errors and misinterpretations. We applaud your critique of scientific papers and acknowledge that no study is perfect. However, it's incumbent on you to accurately represent the papers you discuss and to base your criticisms on facts. In this case, you have not done so. Below is a representative, albeit incomplete, list of the many salient errors that were made in chronological order during your commentary with approximate timestamps. YouTube, we trust that you will correct the record accordingly. So it is too long to read. It would take up another 30 minutes, but I will post the point-by-point commentary. So basically, what they have done at the right timestamps is have our quote and why we were wrong in all cases, basically. And this goes on for many pages. So I'll put it up in the letters section of this episode of TWiV. And I don't know what to say. I mean, I think their points are correct. It was a difficult discussion to have. And I've delayed reading this. I got this letter a long time ago because I just didn't know how to handle it, frankly. But as you can see, if you read the letter, we made a lot of mistakes. But I also think that we got a lot right as well. And so I will chalk it up to a single episode where we got a lot of things wrong, and hopefully we don't do it again. So thanks. Appreciate your writing in.
I have a question about, because they have these timestamps for YouTube. Once you've posted a YouTube, can you go back and add like this list or this counterpoint or anything like that? Or?
That's a good point. I could actually put it in the comments and pin it to the top.
Yes.
Okay, I'll do that. That's fine.
Yes.
And when somebody mentions a timestamp in a YouTube comment, it usually highlights it as a link that people can click, and it goes to that timestamp in the video.
Okay.
Well, I think, Vincent, it speaks very well that you are posting this and reading it and are handling it, that, yes, we take criticism and we want to do what's right.
We want to get things right.
Yeah, I mean, it was a difficult episode, but I have always wanted to get this out here. So there it is. Okay. And I encourage people to go look at because most of our criticisms were not correct. So I encourage people to go look at it if you're interested. So I think that is the way that we will correct the record. I'll put this in the letters, and as Kathy suggested, I will also add it to the YouTube, add to YouTube, pinned to the top. So I can make a comment myself, and I can pin it so it remains at the top of all the comments, and then it's the first thing that people see. Because normally if you
that's a good way.
If you put a comment in, as other people put comments in more recent, it moves down. But as the owner of the page, I can pin it to the top. So
cool.
I'll do that.
Yeah, I didn't even know you could do anything. So that's
yeah, I can put a comment and pin it. So I'll do that. And thank you for writing. I appreciate it. And the commentary is very extensive and shows that we made a number of errors. So thank you for that. And let's do a couple more. Kathy, can you take the next one?
Florencia writes, "Hello, TWiV team. I am quite thrilled to finally be writing to you. I can't say how many hundreds of messages I have written in my head, and also hundreds of questions that I have never asked. I'm a big fan of TWiV, and I constantly seek trusted information and perspectives from this team. I trust each and every one of you with the thorough and honest analysis of data. TWiV is an invaluable resource of knowledge. I have had the extreme pleasure of meeting Vincent when he came in 2015 to give a talk to my institution, Mississippi State University. He had the most spontaneous idea of taking a selfie attached, which I proudly show to anyone that I tell about TWiV. I'm a virologist myself, and I have converted all my students into TWiV fans. I own Vincent's Principles of Virology book, 3rd and 4th editions, a copy of Field's Virology, where I've read Rich's chapter numerous times. Particularly useful is a table that shows the number of infected cells as you increase MOI according to Poisson distribution. In my lab, we have our own wall of herpes, bovine herpes virus 1, made of hundreds of plaque assays. I'm a super fan of plaque assays, and I pride myself in saying that no student passing through my lab will leave without having learned how to do them. I was recently in Barcelona for a sabbatical semester, January to June of 2022, and the daily commuting to the institute where I was working was totally new to me and made me ever so dependent on the TWiV podcast. Now that you are back to one weekly episode plus Daniel's clinical update, I missed the second weekly episode. But I know that you do this out of your own time and hearts, and I understand that you have your own life and jobs too. Having said that, I recently started listening to urban agriculture, and I'm so absolutely hooked. Dixon, my husband and I completely admire you, how much knowledge and life experience you have had, how you tell your stories, blending all kinds of data, facts, phenomena, stories, and people together into a compelling string that keeps us glued to the earpiece. Unfortunately, this means I'm behind with my TWiV episodes. Hey, there's only so much time to walk the dog. But even so, I do not allow myself to jump ahead to newer TWiV episodes. I will listen to them all. Well, you could walk your dog for longer. We heard about a lot of people doing
just
that.
Just get a slower dog.
Yes. I think that I finally decided to write after TWiV 852 when you talked about HPV infection and a rare deficiency in CD28 in 3 individuals, the tree man syndrome. Fascinating episode. I have had in my office for a while, about 14 years now, since I left the University of Nebraska-Lincoln, a little printed comic book named The Curse of the Tree Man. It comes from a website called World of Viruses and gives a link. You probably already know about this site. I'm attaching the PDF file of this particular story about HPV, but there are many other stories, and especially now they have come up with Corona comics, etc. It's worth a look. Maybe this would qualify as a listener pick. Such an honor. With admiration for the incredible work that you do, Florencia. P.S. I've searched for TWiV or Microbe TV on smile.amazon to make donations to your team, but I can't find it. Is there a special tip you can give me? And Florencia is an associate professor at Mississippi State University.
I have found it on the Amazon site. I don't have a problem. Has anyone else?
I did it so long ago, I can't remember the steps.
Let's see. I have to sign in. I don't want to do that.
Maybe we can figure it out, and maybe you can even just post a link to the instructions or something in the middle after her letter or something. I don't know.
2-step verification. Okay, so let's see. There's some part of the thing where it says smile. Where is it? Yeah, I shouldn't do that here because if you, smile. Nope, it's not here. All right, I will make a screenshot of it and post it, Florencia, so you can figure that out. Okay. All right, Dixon, you should read the next one.
Yes, thank you for allowing me to do that. Eric writes, and this is a remarkable coincidence of meeting somebody for the first time and yet knowing who they are. I had the good fortune to quite accidentally meet Dixon in the park this last weekend. We were celebrating, by the way, my grandson's birthday, and his son goes to the same school that my grandson goes to. I really had no idea who he was and was rather enjoying talking about the finer points of angling with him. But at some point in the conversation, he mentioned that he was a professor of parasitology at Columbia. And when I asked him if he knew of any of the folks responsible for Microbe TV, his eyes popped wide open and he laughed, which is true. And all of a sudden it hit me that I was talking about all things angling with the one and only Dixon DePalmieri. Of course, and believe me, I was taken back by his candor of being surprised, etc. He was genuinely moved. Of course, I was rather stunned. You see, Dixon and all of you were my pandemic heroes and heroines. As you might remember, those were dark times back in the beginning of 2020. I'd been trying since mid-January to find kernels of truth in the vast wasteland of information and twaddle we call the internet. And so when I came across This Week in Virology, I felt like I had found a place that was unpolluted by politics, a place that stuck to the science. For that, I thank you from the bottom of my heart. You all performed the most valuable public service imaginable. Through your work, non-scientists such as myself were able to keep ourselves accurately informed, and as such were able to soar just a bit above the nonsense. I felt empowered, in the loop, and armed with the information I needed to keep my family safe. Thank you, thank you, thank you. Before I close this letter, I feel I must touch on 2 points. First of all, I had the good fortune to go to high school with Matthew Pottinger, Trump's deputy national security advisor. And I was with him in Beijing back when he was a journalist for the Wall Street Journal. As a fluent Mandarin speaker, and so is Eric, by the way, Pottinger had provided excellent coverage of the SARS-CoV-1 outbreak. And while we were in mainland China together, he was running down an avian flu outbreak that the Chinese government was vigorously denying despite all the evidence that Pottinger had accumulated. And among the small circle of other government professionals I know, who have had considerable experience working in China, the fact that Pottinger was in the lab leak camp for this SARS-CoV-2 pandemic gave us considerable pause. I am not saying he is right or that he is wrong. I'm just saying he knows China and its government better than most, and that this wasn't his first time at the rodeo. Yes, when you mix science and politics, you get politics. But in China, everything is mixed with politics, including science. And then it is dipped in politics, fried in politics, and served on a bed of politics, with a side salad of politics. And secondly, I would like to formally invite Dixon on a fishing trip, which when I read this email, I just jumped out of my chair. I had been meaning to invite him all through 2020 and 2021, but it's better to be late than never. His timing couldn't have been better, by the way. Thank you all, and forever in your debt, Eric. And Eric, I accept, of course, and thank you for listening and writing into our podcast. Let me tell you what Eric used to do for a living, and I think he's still doing it. Eric is a professional speechwriter. And he's obviously working as a freelancer, and he can't keep up with the requests. And he told me about some of his travel experiences, and the one I was most enthralled with was the one he took to Mongolia, hired by the CIA. Well, I'm going to say the CIA. He didn't say the CIA. He said the United States government's Department of Helping to Spread Democracy Throughout the World. Now, I don't think we ever had a department like that, and neither did he. But he was the only one out in Mongolia doing this work. And that's where he met his wife, and that's where they got married, and they came back to the States, and his child, I'm telling you, this kid, Brendan, is just the most beautiful kid you ever want to see. And he was at the party also because he was helping Ivan celebrate his birthday. And Eric and I probably talked for 2 hours uninterrupted. And the party was going on over there someplace, and we were exchanging information and data and likes and dislikes. And it was really a wonderful experience. And I've never met anybody so intelligent and cogent of what was going on, and at the same time was objective in his thinking and said, you guys do it the best, and that's why I'm sticking with you. You just do it the best. And so the first letter that we read sort of says that we don't do it the best all the time, but we try to do it the best. That's the point. We try, and whenever we do screw up, and that's human, right? We admit it and go on and correct the error and keep going. So, Eric, thank you very much. And he's quite a personable guy, and I really I'm very touched by this podcast association because you don't know who you're talking to when you're on the air. And when people write in, and they write in, of course, and he's been meaning to write for a long time. And I said, whenever you do write your letter, we'll read it. So, Eric, this one's for you, and this is a shout out to Eric, and may you live long and prosper.
Alan, can you take the next one?
Sure. So we're up to John. Here's a question for Rich and Dixon. I know we're supposed to stay in our lane, but nuclear power has me concerned regarding waste. In our many years of nuclear, we've only presented candidates for geographical depositories, of which only one exists in Europe. Some more have been proposed, but we have moved 0 tons of high-level waste to them. That's 7 decades of waste sitting above ground, and there are already energy barons who reaped the profits of these fuels and died before cleaning them up. There was a time until 1973 that countries dumped it into the ocean. Very good, we stopped that. My worry is nuclear power is the ultimate credit card that society doesn't see the interest accumulating on, and I'd wonder what Rich, Condit, and Dixon feel about us leaving that legacy without cleaning it up. They seem to be interested in other sciences and especially particle physics. Shouldn't we ask future reactor builders to commit to a cleanup of previous decades in trade for a license to build more reactors? I just wanted to put this on their radar and would love their input as scientists. John.
Well, John, thank you for assuming that I know all the stuff that we were talking about. So, a particle physicist, I'm not, but I can, and I think everybody on this podcast would agree that if you make a mess, you should clean it up. Yeah. And if you don't clean it up, then there's something wrong with the system, because everybody makes a mess, even in the lab, but we clean it up. And we have mechanisms for cleaning it up. And the nuclear industry has been remiss in a lot of those activities. I can show you an old abandoned Nike missile site that's still hot from fuel that they left. I mean, it doesn't have radioactive material, but it has hydrazine in the soil and things like that. When industry abandons a project and then never comes back to the site, that's the worst crime you can create against humanity. And you don't feel any responsibility for the environment or for people's health or for anything else. You just want to feather your own nest, basically, and you don't want to spend the money to make sure that you don't harm anyone. So that's my attitude, and that has nothing to do with particle physics, by the way. That was a quark. of my own imagination.
Alan, you have any insights?
There's
Alan,
a whole set of So I've tangentially covered related issues on sustainability and such, and nuclear power is one of those things that's just it's so wonderful in so many respects, and the problems with it stem from the fact that it's run by people.
Right.
So there's no unsolvable problem with nuclear. You can fix all this. You can actually reprocess the fuel in a way that drastically reduces the half-life of the resulting waste. You reprocess it into more fuel. It's a great thing to have, that option of recycling the fuel. The problem is that recycling nuclear fuel requires an infrastructure that is identical to the infrastructure you need to build a bomb.
Right.
And so we have this problem that we can't endorse reprocessing because it's part of the system that countries would need to build bombs, and all the disposal options suck for reasons that have to do with humans being humans and being, we don't have the collective social memory to promise that something will actually stay buried for 2,000 years, because we have no idea who's going to be here in 2,000 years. And on and on. It's all of these things that the engineers will say, oh, well, this can be solved this way. And then you look at the situation and say, well, that's not actually going to work in the society that we have. And so, yes, nuclear is undoubtedly part of a solution to dealing with climate change, because we need solutions that can be put into place immediately. But the way we've regulated the industry and the way we're likely to regulate the industry in all developed countries into the foreseeable future means that we're going to have these kinds of problems.
Alan, do you have any insight in the way France handles its problem?
I don't know how they do have a lot of nuclear power, and I don't know.
75% of the country gets its power from nuclear power plants.
And I don't know what their arrangement is for handling the waste,
or
if it's
that the Russians have not destroyed the Ukrainian nuclear power plants. Of course, they expected to take them over, but they realized that if they were to do that, they'd have another Chernobyl on their hands. That's where Chernobyl is anyway. So, we've unleashed this genie that we don't really know how to put back in the bottle, as I heard it expressed by a nuclear physicist once.
Yeah.
So I don't agree with you altogether, Alan, in the sense that I think that nuclear represents a way out of the climate situation. I think hydrogen fuel cells eventually will win, but I'm not sure when that's going to happen.
Until we get a better right. And what I'm saying is, in the near term, we need something.
I realize that
that'll work, and solar and wind are catching up. But anyway, this is far afield from.
So I found 2 articles that are relevant. One from Greenpeace, who claims that France is dumping its nuclear waste in Siberia. Okay, I don't know how that would work, but you could find it. And the other is that they recycle it. Upon its removal from French reactors, used fuel is packed in containers and shipped via train to The Hague. There, the energy-producing uranium and plutonium are removed, separated from the other waste, and made into new fuel that can be used again.
Okay.
So if any of that is correct, who knows?
Except we use cesium rods, though, in the nuclear power plants. It's not uranium, it's cesium, and it's a rod of solid material that you can't get rid of once it's there. That you can't even take it out of the water, otherwise it'll go critical. So, I mean, it's got a lot of really interesting drawbacks.
I'm sure we'll get letters from nuclear engineers.
I hope so.
That's fine. Explain it to us. Yeah.
Yes.
That's all interesting. Anyway, one more email. Brianne, can you read that one?
Sure. Jennifer writes, "Good morning. I listen to the clinical update every Saturday. My dad is in a nursing home in Ohio. Unfortunately, he tested positive for COVID on Monday. He is 77 years old with a history of stroke, chronic kidney disease, diabetes, and high blood pressure. When Mom called me with the news, I immediately asked if he was going to be put on Paxlovid. She wrote it down and called the doctor. He prescribed it, and Dad is starting to feel better. I'm disappointed that the nursing home didn't have a plan in place other than giving him Tylenol, especially because the pulse oximeter showed his oxygen to be 85% and he had a fever of 101. Thank you so much for educating all of us. I may not work in healthcare, but I love the podcast and the information I learned helped my dad. Blessings to all of you, from Jennifer, who is in Kentucky.
Wow. Oxygen, 85%. That's low.
That's bad.
That's very low. That's very low.
Well, I wanted to have this read here because we have so many emails for Daniel, and this one is just a feel-good email that I thought everyone should hear because Daniel always says, have a plan.
Yeah. And so much of the time he has a story that goes the other way.
Yeah.
They called the doctor and they didn't get him on Paxlovid,
Right.
and bad things happened after that. So this is really a good story.
Okie dokie. Time for some picks of the week. And this week, Dixon, we can hear you. You're being recorded. So tell us your pick from last week that didn't make it.
All right, my pick from last week are the vocals, the jazz vocalists. And the jazz vocalists that I have picked as the all-time favorites, at least for me, are the best female vocalists include Ella Fitzgerald, Billie Holiday, and Betty Carter. If you've never heard Betty Carter, you should listen to her. She's remarkable. There's nobody else in jazz that phrases the same song the way she phrases it. And she was a remarkable singer. She really sang from her heart, but also with a unique style that no one else has ever copied or tried to. The best male vocalists include Mel Torme, Joe Williams, who sang with the Count Basie Orchestra for many, many years, and of course the perennial, long-lived man has no telomeres left, and yet he's still living, Tony Bennett. And Tony Bennett has evolved from a crooner in his early days, where nobody except Andy Williams and other people would listen to him. And he's a gifted artist, by the way. Also, he took lots of lessons at the Art Students League. He's multi-talented, and he's a remarkable individual. His phraseology, Frank Sinatra once said, of all the people he's ever heard sing, Tony Bennett is number one in the way he phrases a song. When the songwriter writes a song, Tony Bennett showcases it the best possible way. And Frank Sinatra was a genius at phrasing songs also, but Tony Bennett remains as one of my favorites. And he's done a lot of duets with other people that you might be familiar with, and Diana Krall is one of them, and Natalie Cole, Nat King Cole's daughter.
Lady Gaga.
Lady Gaga.
Yeah, it's great.
We can't leave her out of it. She's fantastic. And he brings out the best in them, and they bring out the best in him, and he's remarkable. So I would listen to anything he would ever sing.
I really like his pairing with young people. I think that's great.
Yeah, me too. I totally agree. Unfortunately, the word on the street is that he's starting to go into a dementia state, which allows him to, if you get him on the stage, he's perfect. If you get him off the stage, he forgets a lot of the things that he used to remember.
He can't remember lyrics, in other words.
Yeah, that's right. He's in his late 90s, as I recall.
Yeah, that's a problem.
Yeah, we should all have that problem. When I get to be 99, I'll let you know.
Well, you're not far off.
Wait a minute. Let's not go there.
Hey, we're here. We're here every week on TWiV.
We are.
Thank you, Dixon. Brianne, what do you have for us?
So I have something that I found out about this summer, and when I thought of the fact that I hadn't picked it, I was sort of like, wait, why hadn't I picked that yet? That's an important thing that I should have told people about this summer when I learned about it. A group of virologists put together some curriculum guidelines for undergraduate and graduate virology courses. So these are the ASV curriculum guidelines. There are a lot of similar things for microbiology courses that ASM has put together. And so this group put together some ideas of what types of things, some objectives things like that, learning goals for undergraduate and graduate virology courses. If anyone listens to Immune, a few months ago, we talked to some people who were making similar guidelines for immunology courses, and so this is the virology version. So anyone who is thinking about designing a course or thinking about updating their course or things like that, this is a really useful reference that I would definitely recommend.
Cool.
Very good.
In addition to when you go to this link on the ASV site, if you go far enough down, it also has a link to the Journal of Virology article that they wrote and published about these guidelines also. So you can even reference that if you want to reference something. Yeah.
Thank you, Brianne. Kathy, what do you have for us?
I picked a video that my younger brother sent me about 6 weeks ago, and I finally had time to watch it. It's only 15 minutes long, but it's about something called the Snow Cruiser, which is something that was built by the United States with funding from the government. Pretty expensive, but it was just ill-conceived and a total failure. But this particular person has put together a really good video about it. And then there's lots of other stories. In his video, he's got a whole reference list and links and things. But it's just kind of amazing. It's this huge thing with these huge, smooth tires. and they built it, I can't remember, I want to say Chicago, somewhere in the Midwest. And then it had to travel east to get on the ship to go to the Antarctic. And so people remember it driving through their community, and it was so wide that they had to close roads so that they could use both lanes and stuff. So, and then because my older brother Bill goes to Antarctic reunions, he met somebody once at one who had salvaged some blank envelopes that were to have been used once the Snow Cruiser reached the South Pole. Of course, it never did. It didn't get anywhere really with those smooth tires and other flaws. But it's worth checking out.
Designed by committee, no doubt.
Yeah.
That's funny. Cool. Alan, what do you have for us?
I have a really cool little news piece. It's from a site called Reasons to be Cheerful, and I think somebody might have picked this site at some point. But now it's in my RSS feeds, and so these types of things pop up. But this is a group of volunteers who are rescuing bats in Ukraine. And of course, you know what's going on in Ukraine right now. Everything's getting bombed by the Russians, and these folks who were involved in wildlife conservation efforts before and were trying to protect bats there for all the reasons that we've discussed on TWiV. This is a very, very important set of species to keep operating, and they're endangered in a lot of places. And these folks are literally running around between bombs to rescue populations of bats that are in buildings that are being destroyed. And the key guy is like set up cages in his apartment to take care of them. It's just this real extraordinary effort.
Wow.
And I thought that was really, really cool.
That's amazing.
Yeah, I mean, you think about all the wildlife being destroyed. Boy,
yeah,
that is interesting.
They also have a cool facility they discuss here that was built before the war. It's in a park that's got a flyway. It's for rehabilitating bats that have been found. And so it's this circular flyway that the bats can fly around and regain their wing strength in. They call it the Bat Collider.
That's funny.
So that's very cool.
That's good. My pick is a PBS video called Woodpeckers, The Whole Story, HOLE. This is just a great video.
I agree.
About woodpeckers, which I didn't know much about. How many different species are there, Dixon? You told me before.
280 some odd species of woodpeckers, and they're found on all the continents, except Antarctica, of course.
This is first. I mean, the photography is gorgeous.
It is,
and they have different species. They talk about how they look for food and how they nest and so forth, what they eat. And my favorite, of course, is the pileated woodpecker, which is the biggest of all and has a huge beak, which it used to hack. I saw one once.
I saw one once here, yeah.
It was just hacking. I mean, it's got a huge reach on its the neck is quite long, and it was just hacking up a log, and pieces of splinters were going all over the place. But it turns out that this bird, every time it hits, it closes its outer eyelid so it doesn't get splinters, right? So coordinated with the hitting. And so the part I really liked was this bird, this woodpecker, the pileated, listens for ants in dead trees and then goes and pokes holes and eats them. He pulls them out, and the ants, they have video of the ants inside frantically running around trying to save things, and this bird just hacks it open and goes in and eats it. Oh my gosh.
It's remarkable.
So anyway, all sizes of woodpeckers, they do different things. The Dixon was saying before, there's one that hides acorns in dead trees.
Actually, they're not hidden. They're in already holes that they've pecked out to look at grubs, right?
Yeah, so they peck out holes and they put the acorns in, they tap them into the...
And they fit them to the size of the hole. If the acorn doesn't fit, they go and get another one until they find one that fits.
Yeah, it's very
They're
cool.
amazing.
There's a fact that I just learned this fall. They must touch on it in this video, that their tongue does this thing that goes and protects the skull. It like retracts over the brain so that they're not getting themselves.
They have a shock absorber built into
Yeah.
their biology. That's absolutely right.
Yeah, I was surprised to see their tongues. I didn't realize they have these very long serpentine tongues. Yeah, they all go in and pull out grubs and so forth. But it's just great, and it's beautifully photographed. You know, it's not easy to do this, and
that's right.
Hats off to them for the video. It's just great.
I love how they land on the log or the tree that their nest is in, because they come in and then they just spread their wings wide open and they softly land, and you see the little chicks with their little beaks come out, and it's adorable. Absolutely adorable.
We have a couple of listener picks. You heard the one from Florencia above, which I will remind you is, this worldofviruses.unl.edu site where they have various PDFs on viruses, including one she talked about on HPV, about the tree man, and also Corona comics and so forth. Really good. And then we also have one from Miguel. It was the individual at the top who was advertising a position over in Houston, and he said, I would like to contribute a listener's pick. The Fold Scope is a paper-based microscope designed to be used in the field and to make research and diagnostics more accessible to remote/underserved communities globally. A link is below, foldscope.com. Thank you for that.
Neat.
Miguel, I've heard of that before. Yep, that's very cool. And that will do it for TWiV 958. You can find the show notes at microbe.tv/TWiV. You can send us a comment, a question, a pick, a correction. If we did something wrong, let us know. No problem. TWiV at microbe.tv. If you enjoy our work, we would love your financial support. We run Microbe TV on your donations. We don't have ads. We don't do any of that stuff. So if you'd like to help us out, we'd love it. Microbe.tv/contribute. Dixon DePalmieri is at trichinella.org, thelivingriver.org. Thanks, Dixon.
It was great.
Glad you made it. Glad you made it to the end this time.
Yeah, me too. So I was just thinking, yesterday we had pumpkin pie and today we had humble pie.
That's fine. I got it. I get it. We make mistakes.
And venison pie.
And venison pie. That's right.
Brian Barker's at Drew University. Still on Twitter. BioProf Barker on Twitter, right?
I'm still on Twitter, mostly because I haven't had enough time this semester to think about any alternatives.
Oh, yeah. Thank you, Brian.
Thank you. It was great to be here.
Alan Dove is at alandove.com. Also, turbidplaque.com. Thanks, Alan.
Thank you. It's always a pleasure.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thanks, Kathy.
Thanks. This is a lot of fun.
Good to have papers back again, right? I'd love papers.
Oh, yeah. Right.
Next week we have an interesting guest, though. Dr. Dan Wilson from Debunk the Funk.
Debunk the funk.
He does a very good job. He's a scientist, and he does a very good job at
I love it.
debunking things that are
The
wrong,
funk.
including many SARS-CoV-2 related things. And he's mentioned us, so I thought we'd get him on and see why and how, right? I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology, the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.